I would like to thank the organizers for giving me the opportunity to be here today to present From Genes to Cognition, Octodondegus, an animal model for Alzheimer's disease translational research. Please, if you are interested, contact our hub, the Degus Hub, which contains today 15 international labs with the aim of understanding this interesting animal model from a multi-lab and multi-perspective approach. So let me take you to the table on the left, where you can see the different characteristics of Alzheimer's disease shared between humans and Degus, such as brain amyloid, tauopathies, cholinergic dysfunction, cognitive deficits, and neuronal death. The Octodondegus is a diurnal rodent and is endemic of Chile, and they live for up to 9 years in captivity, and interestingly, they live post-fertility. So on the right, we can see the different comorbid diseases associated to Alzheimer's in humans and Octodondegus, like macular degeneration, type 2 diabetes, dysfunction in circadian rhythms, etc. Something that is very important to point at this stage of the talk is that not all age Degus will develop Alzheimer's disease. Around 70% of the Degus population will develop Alzheimer's naturally. So this slide is showing you the different behavioral tests on the left that we run on these animals. So we go from the cognitive tests, such as a novel object recognition, T-maze, etc., to the test of daily living. Activities of daily living are defined as the things we normally do, such as feeding ourselves, bathing, etc. And we know that deterioration in the ability to perform these activities is an early sign of Alzheimer's disease. One of the tests among the many in activities of daily living, the borrowing, can detect behavioral changes in AD-like Degus at an early stage between 6 and 12 months old. And this in comparison with the cognitive tests like the T-maze that can only detect impairments between 36 and 60 months old animals. So on the right, I would like to point at panels A and B, and they represent the chronic in vivo 2-photon imaging in apical turfs of layer 5 pyramidal neurons, and they revealed a decreased spine density in AD-like Degus, and this was done in comparison to 5-month-old presenilin-1 mouse model of Alzheimer's disease, delta E9. So these mice, spine loss can be observed in the graph, and also in Octodondegus cortical dendrites, and as you can see in panels C and D, the association between the amyloid plaques to the dendritic spines, as they can point here in the arrows. So the next point is that we ran, so the idea was, is the Octodondegus after all an AD-like natural model? And the Degus genome, we thought, could hold this answer. So we ran a whole genome study, and in 12 Octodondegus from the wild, and this is very important to be mentioned because the colonies from the US and Europe, they come from Degus that arrived to these countries in the 1960s. Therefore, the colonies are highly inbred. So we managed to identify genetic variants associated in AD-like Degus, including APP, MAPT, presenilin-1 and 2, progranulin, TREM1, etc., TREM2, etc. This paper is now under review. Thank you.
Hello, my name is Matías Miaini, and I am a PhD student in Emilio Croft Laboratory in the Leluar Institute, Argentina. We are part of the Degus Hub, and together with patricia.cogram in Chile, we perform experiments to characterize electrophysiologically the hippocampal activity of Octodondegus in vivo. Hippocampus is well known for being involved in memory and navigation, which are impaired in AD. Until now, we only have data on healthy individuals, but we plan to expand this on AD-like conditions. The electrodes goes deep down the cortex and reach CA1 hippocampal subfield. There we can record oscillatory activity. In the left bottom panel we can see an example of this activity. A huge oscillation near 8 Hz can be distinguished, and fast oscillatory activity nested within theta between 30 and 100 Hz called gamma. Spectrogram analysis shows a difference in theta band regarding animals' state. We can see in the right panel, in red, the spectrogram in an open field arena, which compared with the spectrogram in blue at rest shows a peak in theta activity. Electrodes also allow us to record single-cell activity. We are particularly interested in place cells. These are hippocampal cells that fire specifically when the animal is at a certain location. These cells play a major role in hippocampal cognitive map, and it has been shown that degraded representation of space is present in AD models in mice. We found that place cells also exist in Octodondegus. In the left, we can see examples of these place cells. Among the properties of cells, we measure spatial stability, and we found that the majority of cells has higher spatial stability compared with a shuffle distribution of the same data, although only nearly 10% of recorded C1 cells exhibit extreme place cell behavior. Place cells and oscillatory activity are important because of its potential to become AD biomarkers. I would like to acknowledge Cogram's, Croft Labs, Aphraxis, Robert Deacon, Roderick Gigo, and Fondesit, our founding agency, and to thank the organizers for letting us to share our work with you. We welcome new collaborators to the Degus Hub, so please contact us.